So, hi everyone, my name is Patrick Chang from the Curtis Lab in Georgia Tech. Today I'm going to show you that some cells have these polymer cushions underneath the cells, labeled in green over here, and this is a cross-section of side views of the cells. And these cushions can decrease the cell adhesion strength. So first, let's talk about cancer. In cancer, many studies have shown that the increased production of this polymer chain called hyaluronan will cause increase in cell growth and metastasis in various tumors such as prostate, colon, and breast cancer cells. And this polymer chain tethered to the cell surface and form a matrix around the cells. And here is a traditional way people use called particle exclusion assay to visualize this polymer matrix. So here we have a prostate cancer cells, and we put in the particle around it. You can see that the particle does not go all the way to the cell membrane. And this excluded region is the polymer matrix. And this matrix exists on many different cell types, including the chondrocytes here that have its membrane marked in red, and the polymer matrix fluorescently labeled outside the cell membrane. And this polymer matrix is called Pericellular matrix, or PCM. Here we have this green line, the polymer chain that gravitates down the cell surface, and in the matrix, we have this very large negatively charged protein called proteoglycans that can attach to the polymer backbones. And 400 nanometers is very large comparing to the other molecules around. And without these proteoglycans, all the polymer chain will curl up into globule due to entropy. It is this proteoglycans that can stand up all those polymer chain and extend the matrix out, even underneath the cells. And we can see that unlike all the other places, underneath the cells, this polymer matrix form pocket of cushions between the cell and substrate. And now back on the topic of cancer. In cancer, proliferation and migration are very important. And also during those events, the cell adhesion strength need to be weakened in order for them to happen effectively. So understanding how the adhesion strength between the cell and substrate changes is very valuable. And much attention has been given to how these extracellular molecular assembly anchor the cell to the substrate via integrin. And this extracellular assembly has been primarily regarded as the responsible for cell adhesions. But when we take a look at those polymer patches underneath the cells, we see them as a repulsive cushions between the cell and substrate. So we theorize that at the interface between the cell and substrates, it is the repulsive force from this polymer cushions acting with the attracting force from these focal adhesions that can together mediate the complex process of the cell adhesions. To test our hypothesis, we are taking this two-man route. Applying a controlled hydrodynamic shear to cells with different PCM conditions can provide us a mean to quantitatively measuring the effect of PCM to the cell adhesion strength. And the other route is looking at the spatial correlation between the PCM and the focal adhesions. It's the first time people use super resolution microscopy to look at the two assembly underneath the cells and see if they are spatially exclusive to one another. Today, I'm going to focus on the quantitative measurement of the adhesion strength. To do that, we want to apply a control shear to the cell. The stronger the cell adhere to substrate, they can withstand a higher shear before detaching. In collaboration with the Garcia lab in Georgia Tech, we are using this so-called spin disc apparatus to measure the cell adhesion strength on thousands of cells under different shear conditions in a single run. And this method is well established to quantifying those cell adhesion strength. To do that, we first let the cell adhere, and then after it's submerged into the liquids and spin by the motor, all these cells will feel a shear strength that's proportional to the distance to the center of rotation. That is, on the surface, all the cells will, at different positions, will feel a different magnitude of shear, with there's no shear in the middle and a higher shear at the edge. After the spin, I count the cells remain on the surface along the radius, and the higher the shear, the less cell will remain. And with the highly studied hydrodynamic of the spinning disk, I can relate the position of the cells to the shear stress they felt on the surface. And so we can then plot out the relationship between the shear stress and the fraction of the cell remain on the surface, and use the use a function to find out the adhesion strength, or the tau 50, which is the shear stress needed to detach half of the populations. So now I'm going to show you some of our preliminary data on the cell adhesion strength. Here is a data fit of our control cells, and this bar chart shows the adhesion strength of the standard PCM. After the cell is well adhered to the substrate, we'll apply an enzyme called hyaluronidase to chop off all the hyaluronan backbones in the matrix, and then we measure the cell adhesion strength. It shows us that the adhesion strength increased small, but then statistically significant amount. And on the separate samples, we use another enzyme called conjoined ABC to chop off all the proteoglycans inside the matrix, and we measure the adhesion strength to be a lot bigger, more than doubling the adhesion strength of our control cells. At first, you might be wondering why chopping off those decorating proteoglycans that increase the adhesion strength so much more than getting rid of the polymer backbone of the matrix. That was surprising for us at the beginning too. But when we take a closer look, underneath the cells, all those PCM are formed in patches, and we suspect that when we degrade those polymer backbones, all those very large negatively charged proteoglycans still trap underneath the cells and still form a repulsive cushions between the cell and substrates. But when we degrade those proteoglycans, there's nothing else to hold up all those polymer chains, and then they curl up into small globules, and thus decrease those repulsive force acting as of the cushions, and thus increase the overall adhesion strength. So here, I provide some evidence that showing the mechanical function of our matrix that can decrease the adhesion strength. In conclusion, I provide the preliminary results of the PCMA acting as a mechanical repulsion cushions that affect our cell adhesions. In the future, we want to continue working on the cell adhesion strength versus the PCM conditions, particularly using genetic modified cells with tunable hyaluronan polymer chain, the proteoglycans, and even the CD44 receptor on the cell membrane. We also want to continue to use the super resolution microscopy to like the spatial distribution of the PCM and the focal context. Also want to determine all those PCM-mediated adhesion. Are they mechanical as we theorize, or they are via signaling? And overall, we want to focus on cancer, and especially the role of PCM on the cell cancer metastasis. At the end, I want to thank my PI, Jennifer Curtis, my group, my collaborator, funding, and post-network for all the support and opportunities. And thank you all for listening.
Setting up, we have time for one quick question. Patrick?
I know you didn't talk about this, but I mean the issue of the spatial organization of the focal adhesions versus the matrix material is even more interesting when the cells are moving, because then you have to form new focal adhesions. So are they likely to form regions that they have to push the matrix away? And you have some vision as to how this might work? I know you may not have any data yet, but you have some idea about how it might work?
Yes, we think that during the migration, the front sides of the matrix will have less of the matrix, so then that can adhere better. But then the backside will have a thicker coat, thicker cushions underneath the cells, so then the adhesion is weakened, so then they can move forward, drag the cells forward. That's how
You're trying to show this experimentally, I guess?
Yes, we are going to look
Okay,
into it.
great.
Okay, to keep things moving along, why don't we thank Patrick again?